In the late summer of an archaeological season that was expected to be routine, a team of researchers working at a windswept excavation site uncovered a human skeleton unlike anything they had planned to find. The region had already been studied for decades. Layers of soil had been cataloged, pottery fragments analyzed, and settlement patterns mapped with confidence. By all expectations, the bones beneath the ground should have belonged to people whose ancestry and culture were already well understood. But this skeleton did not behave like the others. At first glance, nothing appeared unusual. The burial position matched local traditions. The grave goods were minimal. The surrounding soil dated to a period historians believed was fully explained. Yet small details began to raise questions. The proportions of the skull were slightly atypical. The shape of the jawline did not match known populations from the area. Even the wear patterns on the teeth hinted at a diet that seemed inconsistent with what archaeologists believed people there had eaten. The discovery might have remained a minor note in a field journal if not for one decision. The team chose to extract samples for genetic testing. Ancient DNA analysis has transformed archaeology in recent decades. Where earlier generations relied mainly on pottery styles, architecture, or written records, modern researchers can now compare genetic sequences and trace migration patterns across thousands of years. Bones once viewed as silent artifacts now carry biological records that reveal ancestry, movement, and relationships between populations. When the sample was sent to the laboratory, expectations were simple. The DNA would likely confirm what the archaeological layer already suggested, that the individual belonged to a known regional group. The result would strengthen current theories and help refine timelines. Instead, the preliminary analysis produced a warning. The genetic markers did not match the expected profile. Technicians initially assumed contamination. Ancient DNA is fragile, and even small exposure to modern material can distort results. The sample was cleaned again, controls were repeated, equipment was recalibrated. A second extraction was performed, then a third. The result remained the same. The skeleton carried genetic signals linked to a population believed to exist far away, both geographically and chronologically. According to accepted historical models, there was no known pathway that could explain how such ancestry appeared at this location during that period. News of the anomaly moved quietly through academic circles. Researchers are cautious by nature. Extraordinary claims require extraordinary evidence, and premature announcements can damage credibility. The team chose to remain silent until further testing could confirm or challenge what they were seeing. Meanwhile, archaeologists revisited the excavation site with new attention. Soil samples were reanalyzed. Carbon dating of nearby organic material was repeated. The burial context held firm. Every line of evidence suggested the skeleton truly belonged where it was found. There were no signs of disturbance, no evidence that the body had been moved from another era. The contradiction deepened. If the dating was correct, and the DNA was correct, then at least one of the assumptions underlying regional history would need revision. Some scholars proposed a straightforward explanation: long-distance migration. Humans have traveled farther than traditional models once allowed. Trade routes, climatic pressures, and social upheavals often push people into unexpected regions. Perhaps this individual represented a rare traveler, an outsider who died far from home. But the bone chemistry complicated that theory. Isotope analysis, which can reveal where a person grew up based on water and food sources recorded in their teeth, suggested the individual had lived locally for most of their life. This was not a stranger passing through. The person appeared to have been raised in the area. How, then, could local upbringing coexist with distant genetic ancestry that historians believed should not yet exist here? As the team expanded the investigation, they began comparing the skeleton to other remains stored in museum archives. Many collections had been excavated decades earlier, before DNA testing was possible. Quietly, samples from several older finds were re-examined. A pattern began to emerge. One skeleton could be dismissed as an anomaly. Two might still be coincidence. But multiple individuals carrying similar genetic signals suggested a larger story, one that had gone unnoticed because earlier researchers lacked the tools to see it. The implications were uncomfortable. Historical narratives often assume gradual and predictable movement of populations. Civilizations are mapped like steady expansions on a timeline, but genetics sometimes reveals sudden shifts, hidden migrations, or forgotten connections that leave little trace in written records. Some historians cautioned against dramatic conclusions. DNA alone cannot explain culture, language, or identity. Others argued that the findings hinted at a migration event never recorded in surviving texts, a movement so early or so small that it left almost no material evidence. As debates intensified, the identity of the individual itself came back into focus. The skeleton showed signs of a physically demanding life. Muscle attachments suggested repetitive labor or travel. Minor healed injuries indicated survival through hardship. Whoever this person was, they lived in a world more interconnected than modern historians had imagined. The research team prepared their first internal report. The language was careful, almost conservative. Terms like unexpected ancestry, preliminary interpretation, and requires further study dominated the text. Yet between the cautious phrases, a quiet realization was forming. The discovery challenged the central assumption about who lived where and when. When rumors of the findings began to spread beyond academic circles, reactions were mixed. Some saw exciting evidence that ancient human history was more complex than textbooks allowed. Others warned against sensationalism, pointing out that science advances through gradual accumulation, not dramatic headlines. Still, the question refused to disappear. If this skeleton truly belonged here, then history contained a gap, a missing chapter. A movement of people that had slipped through written records, surviving only in the DNA of the dead. The team decided to expand testing using newer sequencing technology capable of reading degraded genetic fragments with greater accuracy. The results would take months. Funding applications were submitted. Collaborations were formed with international labs to ensure independent verification. As preparations continued, one senior researcher reportedly summarized the situation with unusual bluntness. "We thought we knew who lived here." The bones are suggesting otherwise. For now, the skeleton remained in controlled storage, carefully preserved, heavily monitored, and at the center of a growing scientific mystery. It was no longer simply a set of ancient remains that had become a question directed at history itself. And the deeper researchers looked, the more they realized this discovery might not be an isolated case, because the DNA results that shocked the team were only the beginning. By the time the third phase of research began, the discovery was no longer viewed as an isolated curiosity. What started as a single skeleton had grown into a larger scientific investigation, involving geneticists, archaeologists, climate scientists, and historians attempting to answer a question that grew more complex with every new result. How many chapters of human history remain invisible to traditional evidence? The team returned to the original excavation site with a different mindset. Early work had focused on confirming the anomaly. Now the objective was reconstruction, identifying when the unexpected ancestry first appeared, how long it remained, and what eventually happened to the people who carried it. Deeper layers of soil were opened carefully, each level representing a step further into the past. As additional burials emerged, researchers documented every detail with extreme precision. Unlike earlier generations of archaeology, nothing was considered too small to record. Soil composition, pollen traces, microscopic residues, and bone positioning were all treated as clues in a larger puzzle. DNA extraction began again. The first new results arrived quietly. A skeleton from an earlier layer showed partial genetic overlap with the original individual. Another had weaker signals but still indicated distant ancestral links. The pattern suggested something gradual, not a sudden invasion or dramatic replacement, but slow integration over time. This interpretation changed the tone of the discussion. Instead of imagining a mysterious group arriving and disappearing, researchers began to see a scenario where populations blended subtly across generations. Migration, they realized, does not always look like conquest or mass movement. Sometimes it resembles a steady flow of people, marriages, and shared survival strategies that leave minimal traces in material culture. Climate research added another piece to the story. Data, reconstructed from sediment cores and ancient plant remains, revealed a period of environmental instability during the era in question. Shifting rainfall patterns may have altered landscapes, opening corridors that connected regions normally separated by harsh terrain. For a limited time, movement between distant populations may have been easier than historians previously believed. If so, the DNA evidence was not impossible at all, only unexpected because earlier models underestimated how dynamic ancient environments could be. Still, the implications remained profound. Textbook history often presents ancient populations as stable and geographically fixed. Genetics increasingly suggested the opposite. Humans were moving, mixing, and adapting continuously, even in eras once assumed to be isolated. The boundaries historians used to divide cultures might be modern simplifications rather than realities experienced by people living at the time. As the team prepared their broader analysis, they faced a challenge beyond science: interpretation. Public interest in ancient DNA had grown rapidly, but genetic findings are easily misunderstood. Ancestry does not define identity, and biological connections do not automatically imply shared culture, language, or belief. The researchers emphasized this repeatedly, careful not to allow sensational narratives to overshadow nuanced conclusions. Yet one question lingered among the team itself. Why did the genetic signal eventually disappear? Later burials from the same region showed decreasing traces of the unusual ancestry until it faded almost completely. There was no sign of violence, no evidence of sudden collapse, and no clear replacement population. The most likely explanation was assimilation. Over generations, lineages blended into larger communities until distinct markers were diluted beyond recognition. In simple terms, the people did not vanish, they merged. This realization reshaped the emotional tone of the project. The skeleton that once seemed to represent an impossible outsider now appeared as part of a broader human story. Movement, adaptation, and quiet integration that written history never recorded. The final analysis combined genetics with archaeology, climate modeling, and migration simulations. The conclusion was careful but clear. Human interaction across regions had likely occurred earlier and more frequently than historians assumed. The evidence did not rewrite all of history, but it exposed gaps, places where existing narratives were incomplete. Academic reaction was mixed, but increasingly supportive. Some scholars argued the discovery confirmed trends already hinted at by newer research, while others emphasized the need for more data before revising timelines. Debate remained active, as it should in science. Every new answer produced further questions. Museums began re-evaluating old collections. Skeletons excavated decades earlier were now viewed differently, no longer just cultural artifacts, but potential genetic archives. Projects that once seemed settled reopened for analysis. What other unexpected ancestries might be hidden in storage rooms, waiting for technology capable of reading them? The original skeleton, however, retained a unique significance. It represented the moment when assumptions collided with evidence, when history shifted from certainty to inquiry. The bones themselves did not speak, but the data they carried forced researchers to reconsider how fragile historical narratives can be when built without biological evidence. In later interviews, members of the research team reflected on the broader implications. One point appeared repeatedly: history is not a finished book. It is a reconstruction shaped by available evidence, and when new tools emerge, the story changes. Ancient DNA was not replacing archaeology or historical scholarship, but expanding them, revealing layers previously invisible. For the wider public, the discovery carried a deeper message. Human history is far more interconnected than modern categories suggest. People moved, met, and mixed across vast distances long before written records began documenting such interactions. The lines separating civilizations may have been less rigid than once believed. As the project concluded, researchers archived their data and published findings openly, inviting others to challenge or expand the interpretation. Science moves forward through scrutiny, and the team knew future discoveries might refine or even overturn parts of their conclusions. The excavation site was eventually closed again, returning to silence. Wind passed across the surface where generations of lives had once unfolded, leaving almost no visible trace. Beneath the ground, countless stories remained buried, waiting for future questions. The skeleton that started it all was preserved under controlled conditions, no longer seen as an anomaly but as evidence of a larger truth. History often hides its complexity not because it is lost, but because we lack the tools to see it. And perhaps the most important realization emerged only at the end. The question was never simply who this individual was. The real question was how many more impossible discoveries are still waiting, quietly resting beneath the earth, ready to challenge everything we think we know about where we came from. Months after the first unexpected DNA results, the research team gathered again, this time not at the excavation site, but inside a genetics laboratory where rows of sequencing machines quietly processed fragments of the ancient past. The goal was simple: verify the anomaly beyond any reasonable doubt. If the earlier results were correct, the implications for early human migration patterns would be significant. If they were wrong, the case would become another example of how fragile ancient DNA research can be. The first step was replication. Independent laboratories in different countries received blinded samples, meaning researchers analyzing the DNA had no information about the skeleton as origin or the expectations surrounding it. This method is standard in high-level research, designed to eliminate bias. If multiple labs reached the same conclusion, the likelihood of error would drop dramatically. Weeks passed. Then the reports began arriving. The outcome was consistent across institutions. The genetic profile remained the same. The skeleton carried ancestry markers linked to a population historians believed had no documented presence in that region at the time. More importantly, the data showed clear signals of admixture, a blending of lineages, suggesting that this individual was not a random outsider, but the result of generations of interaction. This single detail changed the discussion completely. If ancestry mixing had occurred, then there must have been sustained contact between groups long enough for families to form and descendants to live locally. Such interaction leaves traces, shared tools, borrowed techniques, mixed burial practices. Yet the archaeological record showed little evidence of this. The contradiction deepened. To understand how unusual the findings were, the team compared the skeleton's DNA to thousands of ancient genomes stored in international databases. Genetic similarities appeared in places separated by vast distances. These connections were statistically significant, but historically inconvenient. They suggested movement or communication across routes that traditional models considered unlikely or impossible for that era. Some researchers proposed an early migration wave that had gone largely undetected. Climate changes during the period may have opened temporary corridors between regions. Rivers shifted. Grasslands expanded or collapsed. Human groups, driven by survival, may have traveled farther than expected, leaving only faint traces that archaeology had not yet recognized. Others were more cautious. Genetics reveals biological relationships, not necessarily cultural identity. A person can inherit ancestry from distant populations without representing a large migration. Small groups sometimes move, integrate, and disappear culturally within just a few generations. Perhaps the skeleton represented one of these subtle events, too small to reshape material culture but large enough to leave a genetic footprint. The debate intensified when isotope analysis was revisited with improved precision. The chemistry of the individual's teeth confirmed earlier results. The person had grown up locally. Seasonal patterns recorded in the enamel matched environmental conditions of the region, not distant lands. This meant one thing clearly. The unexpected ancestry had been present in the area long enough for children to be born and raised there. Researchers began examining older skeletons from nearby sites that had been excavated decades earlier. Many were stored in museum collections, labeled according to assumptions made before genetic tools existed. Small samples were taken under strict ethical guidelines and sequencing began. The results shocked the team again. Several additional individuals showed similar genetic signatures. Not identical, but related. What initially appeared to be a single anomaly was now becoming a pattern. Historians started asking new questions. If these populations had interacted, why were there no clear records? Written sources from the era were limited but not absent. Trade networks were known. Certain cultural practices were documented, yet none mentioned a group fitting the genetic evidence. One explanation emerged. The communities may not have considered themselves separate at all. Cultural identity often changes faster than genetics. A population can adopt local customs, language, and burial styles within a few generations, blending into surrounding society while still carrying distant ancestry. In other words, the people may have vanished culturally while remaining biologically present. As findings circulated at conferences, reactions ranged from fascination to skepticism. Some scholars warned that genetic data was being interpreted too aggressively. Human history is complex, and isolated findings can sometimes exaggerate patterns that later disappear with broader sampling. The research team responded by expanding comparisons further, integrating archaeological data, climate reconstructions, and migration models built using computer simulations. When these fields were combined, an intriguing possibility emerged. A window of environmental change may have created a temporary rot connecting regions previously thought isolated from each other. If true, this would explain why the genetic signal appeared briefly and then faded. The root may have closed again due to climate shifts, cutting off contact and leaving only descendants behind. Yet, not everyone accepted this interpretation. Some archaeologists pointed out that tools and artifacts at the site showed little foreign influence. If new groups arrived, why didn't their material culture spread? Others argued that social boundaries might have limited exchange of technology while still allowing intermarriage, a scenario difficult to prove through artifacts alone. The skeleton itself remained central to the discussion. High-resolution scans revealed healed fractures suggesting a physically demanding life. Bone thickness indicated regular movement across uneven terrain. This was not an elite buried with status symbols, but an ordinary individual, someone whose existence would normally have disappeared into history unnoticed. And that was exactly what made the discovery powerful. History is often reconstructed from rulers, monuments, and written records. Genetics reveals the lives of people who left none of those things behind. As researchers prepared a public paper, language became increasingly cautious. Claims were framed in probabilities rather than certainties. Scientific integrity demanded restraint. Still, the conclusion was unavoidable. Human populations in this region were more diverse than previously believed. The publication triggered global interest. Headlines emphasized mystery and controversy, though the researchers themselves focused on measured discussion. Interviews highlighted uncertainty, stressing that the discovery raised questions rather than providing final answers. Within academic circles, new projects began forming. Excavations previously considered low priority suddenly gained importance. Museums reopened archives. Funding agencies allocated resources for broader ancient DNA surveys. What started as a single skeleton was reshaping research priorities. Yet, despite the progress, one mystery remained unresolved. How widespread was this hidden ancestry? Was it a small local phenomenon, or evidence of a much larger migration event still buried beneath centuries of assumption? As sequencing technology advanced, the team prepared to test deeper layers at the original site, older burials that might reveal when this genetic signal first appeared. Because if earlier skeletons carried the same markers, then the story would reach further back in time than anyone had imagined. And that possibility threatened to challenge not just regional history, but the entire timeline of human movement across the ancient world.